Introduce myself. I'm Leonard Foster. I'm in the Biochemistry and Molecular Biology Department, also the Michael Smith Labs, and also the Life Science Institute. So today, I'm not going to tell you any biological stories. I'll tell you some hints of biology, but I want to, as my title says, give you a bit of an update on where these new fields of single-cell proteomics and lipidomics are at least in our hands, and what some of the steps we've gone through to get to be able to do what we are doing, and some of the outlooks for what one might be able to do with these technologies in the future. So I always start off by thanking the people who did the work, and we have a larger group than what is shown here, but I think everybody who's been primarily involved in the work is shown here. So Lucy has really been championing the single-cell proteomics and then also some lipidomic work. She has worked a lot with Jason on the proteomics side and Armando on the metabolomics side to get things working. And then Osai has also jumped in a little bit. Some other work on the more informatics side has been being led by Juan, but I won't really talk much about that. And our funding has come from several different sources for this. Okay. So everybody's probably aware of single-cell RNA-seq and sort of how much that technology has changed our view of biology and our ability to probe variation at the single-cell level and pick out the effects of or the differences in what might be going on within subpopulations of particular cells. And it's found application all over the place. I'm not really going to drill into what all the parts of this picture show, but it really allows you to, like I said, pull apart subpopulations of cells and characterize them differently, or characterize how they're different at the level of transcriptional control, looking at how cells might migrate, not so much physically moving, but how their transcriptional program might change during development, and all sorts of other really cool questions. For a number of years there, it seemed like you couldn't open a journal or an issue of Science or Nature without having at least one single-cell RNA-seq paper in there. So that's all great. And for those of us in the later, or focusing more on the later steps of the central dogma, it was yet another case of feeling like we were getting left behind by the sequencing field. The first sort of instance of getting left behind, I think, was at the genome sequencing level, where you were able to sequence whole genomes, look at whole transcriptomes in bulk samples, and proteomics was left looking at like 15–20% of, in principle, of what was expressed in any given cell. And proteomics technology has come a long ways now. We can relatively easily find 9,000 or 10,000 proteins in a particular cell type if we put a bit of work into it. The RNA sequencing allows you to look at more, probably 12,000 to 13,000 transcripts pretty reliably. But anyway, proteomics was catching up there. But proteomics then quickly got left behind again with the single-cell technologies. But again, I think we're catching up again. The later omics, the metabolomic technologies have lagged even behind proteomics. So those are also catching up. But I put this little graphic up here as a good reminder for why it's helpful to be able to actually look at the protein level. So this is some data that our group, mostly led by Jason and Renata in the core facility, collected with Jim Johnson's group and a few other groups around the country This particular data set is from islet cells looking at protein abundance on the y-axis and RNA abundance on the x-axis. And if you were challenged by someone, they said they'd give you the level of an mRNA, a transcript in those cells, and your job was to come up with what the protein level is, you would probably not want to bet a lot of money on your ability to accurately predict that. So this has been seen over and over again. The levels of transcripts correlate very poorly with the levels of proteins. And so if you're using just the transcripts to try to predict or understand something about the biology, you're missing quite a lot. And this doesn't even take into account the products of the proteins, which are the metabolites. Anyway, this is what we in the proteomics field keep telling ourselves about why it's worthwhile continuing to look at proteins. So I got into the proteomics game in the early 2000s. The field was really just taking off. This was during my postdoc, and you don't really recognize it when you're in the middle of it. But looking back, it was clear what was going on in the field at the time. Like, as I said, it was new. Everybody understood that. But there's this numbers game race. Who can identify the most proteins? Every person in any given lab that was specializing in this would be doing their own methods. There was no sort of consistency lab to lab, or even within the lab. And the best practices for everything from how to handle samples to how to analyze the data didn't exist. And so there was a lot of disagreements. There's a lot of sort of false starts and people promoting methods that turned out to be kind of crap in the end. But you don't really know that until you're until you can look back afterwards. But with that sort of experience, it really feels like we're going through the same thing again with single cell proteomics. And I know single cell RNA-seq has gone through the same thing as well. But there is very little agreement right now on the best ways to handle samples. The actual use of the mass spectrometer is probably somewhat standardized. Everybody kind of does maybe their own little tweaks, but overall those are probably pretty consistent. But then how you analyze the data is very much up in the air as well. And again, I've mentioned proteomics here a few times, but the same thing, it really applies to at the level of the metabolomics as well. So I'm going to tell you about how we've tried to deal with some of the problems, how we've tried to, how Lucy really has had to basically develop everything herself from scratch, because in a lot of cases, the way labs have been able to get some of these things done initially was by developing some kind of custom hardware or custom software that isn't easily reproduced in another lab. And I would say we're kind of guilty of that as well. We didn't develop something ourselves, but worked with another group that did, and I'll mention a bit about that. But the exciting thing for us is that the progress, both for proteomics and lipidomics, has been really exciting, and I think it allows us now to ask and answer some biological questions that were not really, you couldn't even think about asking before. Okay, so these are the kind of steps I'm going to break my talk down into. I won't talk. I'll talk to different degrees of depth on each of these things, but I'll try and touch on everything. So, but actually maybe not so much about getting cells. Getting the cells, you have to be able to get samples yourself or collaborate with someone, but choosing the right sample turns out to be key. So in single cell proteomics and metabolomics right now, one of the rate limiting steps is the mass spectrometry time that you need to analyze one cell. And so what that means is that in a week, for example, we could analyze several dozen cells, maybe let's say 60 to 80 if things are going well, maybe more again if everything's really working perfectly, but we're in that range. Whereas a single cell RNA-seq sample will look at many thousands of cells in sort of the same time frame. So we're at least an order of magnitude away from RNA-seq in terms of throughput for cells. And that has a big impact on the biological questions you can ask, because if you are looking at subpopulations of cells that might be present at 1 or 2%, you're going to need to look at like at least 1,000 cells, probably considerably more, to really get good sampling of that 1 or 2%. And if you can only look at 100 cells, yeah, you probably shouldn't do the experiment. And so that's kind of where we're at, and where some of the biggest growth probably is for single cell proteomics and metabolomics in the future. So that sort of governs the kinds of questions you can ask. But if you're looking at things that that might be present at 5 or 10%, then for sure you can do these kinds of experiments. But anyway, you have to get the cells somehow. And then you have to get the cells into a well. So there's a few different technologies that are used for RNA-seq for extracting RNA from individual cells and barcoding it or whatever you choose to do with it. But it turns out most of those methods aren't compatible with a mass spectrometer as a detector. They work well for a sequencing platform, but not so much for a mass spectrometry platform. So not quite all, but most of the things that have been developed for RNA-seq in terms of sample handling don't really translate over. So we had to come up with new approaches to doing these kinds of things. So if you want to put individual cells into a well of a 96 or 384 well plate, there's a number of ways you could do it. So we tried flow cytometry at first, and Lucy didn't have much success there, mostly because the device didn't seem to be very good at actually counting the number of cells that went into a well. So you might get anywhere from 0 to 20 cells, where really you want 1 cell or maybe no cells optimally. Lucy's since gone back and had a bit more success there. So flow cytometry is one option, but it's not really the one we use all that much. You can also imagine manually manipulating cells somehow, laser capture microdissection, or just pipette or other kinds of devices. This is relatively low throughput. I've put electrophysiology up there under that line. And the reason for that is one area where manually putting cells into a well makes sense is if you're making electrophysiological recordings of individual cells. There, your rate limiting step is not how long it takes a human to put 1 cell into a well. It's how long you're recording whatever it is you're recording about those cells. And so after you're done recording, then you can move that cell into a well, and you might do a dozen cells in a day if you're really good. But getting the cell into the well is not the rate limiting step. So we are working with a couple different groups to analyze cells where there's been sort of deep characterization of individual cells at the electrophysiological level. And then we can hopefully look at the proteins and maybe how those are different relative to the electrophysiological outputs of those cells. So that's one option. You can imagine manually diluting a suspension of cells and then taking like 1 microliter and putting into a well. If you've ever tried that, you'll know it doesn't work very well. At that level, you get a binomial, at best, a binomial distribution of either 0 or 1 cell in your pipette, and you don't really have a good way of figuring out whether there's a cell in there or not. So that's really not very useful. Then you come to microfluidics and sort of very closely related inkjet printing technologies. And I'll tell you a little bit more about that. So this device was the first one that we kind of got acquainted with. It was developed at a university in Oregon, and Armando, through his previous work, connected us with the lab, or one of the labs that was involved in developing it. It's now marketed by TCAN, was initially developed by Hewlett Packard, and it's based on inkjet printing, as you might expect, coming from Hewlett Packard. And basically, it allows you to put a droplet into individual wells, and it's got a mechanism for determining whether one cell was successfully deposited or not. Importantly, it then also allows you to add other reagents, so it acts as a liquid handler in that way. But it's working at the sort of microliter level, and that becomes important later on. But it is a pretty reliable system, and so if you can get cells into suspension, then we can put them into individual wells. There's some restrictions, like really big cells and really small cells don't seem to work all that well, but overall it works reasonably well. So then, oh, what is that? Okay. Somehow the acknowledgement on this slide is not showing up. So we were talking with various people around the city, and Karen Chung's group in School of Biomedical Engineering was developing a single cell device as well. That was originally developed in collaboration with the Genome Sciences Center, I believe, to develop single cell RNA-seq and other genomic-based methods. But they were interested in trying to apply it to proteomics experiments. And so we're working with Eric in Karen's lab to try to apply this to do single cell RNA-seq. So on the left is the device I just told you about, the TCAN device. Eric and Karen call their device the Isolatrix. Effectively, it works in a similar way using sort of inkjet printing approaches, but it's working with much smaller volumes. And again, that becomes important. So down to the, I think 50 picoliter, picoliter. Sorry, must be nanoliter, not picoliter. 50 nanoliter volumes. 500 picoliters. Okay, thanks, Robin. This again is putting there's a cell in a droplet that is sort of spat into each well. The important thing is it's a much smaller volume than what the Isolatrix, the UNO device can output. And so how that manifests, so what Lucy did was work with Eric to basically use this device to put cells into wells, use the UNO device to do things in parallel, and then ask the mass spectrometer what do the data look like. So the data on here are the data I'm showing are looking at two different cell types. These have been Lucy's favorite cells to work with so far. A549, which is a pretty commonly used cell line. And then Lucy's PhD project has been focused on astrocytes. So we've been looking at male and female astrocytes as well. And the main thing, I need to squint here myself to see this. So there's two different donor astrocytes in this particular experiment, or two different astrocytes from two different donors, I should say. And on the y-axis in the upper left here are the number of protein groups. Each dot represents a different cell. And the main thing to take away here, I guess, is that we're averaging about 2,600 proteins identified and quantified from each individual cell. And some are less and some are more, of course. But that number puts proteomics right about where single cell RNA-seq is at. So in a sequencing or in a lane of a sequencing device, you've got so many reads that you have to divide amongst all of your cells. With barcoding, you can put multiple cells or the RNA from multiple cells into one lane. And the way people do these experiments, they're adjusting things so that they end up getting quantitative data on roughly 2,000 transcripts in a cell. So at 2,500 or so proteins, we're getting at least as many, if not more, proteins identified and quantified in these cells as you would in an RNA-seq experiment. Again, the caveat is that we can't analyze nearly as many cells at this depth as one could with RNA-seq. But anyway, I'll skip ahead to this slide actually. So, because this is really showing the comparison between the Isolatrix device and the UNO device. So again, if you probably can't read this all that well, but the first two bars here are the A549. This is the data from donor 1 of astrocytes and donor 2. I'll get to the sex of these donors later on, but that doesn't matter so much for this particular experiment. And then you've got this pair of different sets of data. The first one is the data coming from the Isolatrix. The second one is the data coming from the UNO. And so reproducibly over and over again, we're always getting more proteins identified from cells handled by the Isolatrix compared to the UNO. And that we feel comes down to the just a concentration effect. The Isolatrix is putting out a much smaller droplet. All the biochemistry that one needs to do to extract proteins and digest them is happening in a smaller volume. You're getting more efficiency that way, and therefore getting more proteins identified at the end. Okay, so how well or how reliable, I guess, are the data? So this is a tough question to really answer because we don't know what the ground truth is. There's no like gold standard. You have to compare everything to this. But what we can do is for any given protein identified, compare the expression level in one device versus the other device. And overall, the correlation is pretty good. A given protein measured in one device correlates pretty well with the same protein in the other device. The problem is those are not the same samples, right? You really can only analyze one cell here and a different cell there. So you have to kind of hope that at a sort of epidemiological level that you've got some consistency between the two devices. But it seems like you do. Okay, so that's getting the cells into the wells. How do we extract and digest the protein? So again, a lot of the conventional methods that one uses in proteomics don't really scale down to this level all that well, mostly because it'll lead to too many sample losses. All the manipulations you can do with a large amount of protein, you don't really care if you lose 10% of the protein. Because you've still got lots left. But at a single cell level, that becomes much more critical. So what Lucy has worked out, and again, things kind of change a little bit all the time, but one great way to lyse cells is freeze thawing. And you don't really, it's sort of loss free. You might lose a bit of water, but you don't care about that. But you're not passing the sample through different pipette tips or whatever, and losing material that way. So you can freeze thaw samples. For the trypsin digestion, if any of you have done proteomics before, you'll know that we usually reduce and alkylate all the cysteines. That helps block the really reactive sulfhydryl groups, and overall generally gives you better results in the end. But that all those extra steps would lead to more losses. And so we've realized that we really don't need to bother with that. You're not going to probably find a few of the cysteine containing peptides because of that, but you lose less of the other things by not doing it. So it makes more sense. Another thing you don't usually do in proteomics is add detergent into your sample that's going to go into a mass spectrometer. But there's one particular detergent that is used, was used well before single cell proteomics came along, but it's considered to be mass spec friendly, whatever that means. But it doesn't actually seem to lead to the same problems that many of the other detergents lead to. But this is endodecyl beta D maltoside, or DDM. So we add a bit of DDM in. The main thing it probably is doing is stopping peptides from adsorbing to the plastic of whatever sample handling plates you're using. And another big factor is that at these extremely low volumes, evaporation is a huge issue, right? So you have to try to do all of this under very high humidity to prevent evaporation. And also we do things faster than we normally would in bulk samples, like with shorter digestion times, that kind of thing. Okay, then the instruments, I won't talk too much about this, but we've been fortunate to get a new instrument called the Ultra 2 into our proteomics facility, the one over in Michael Smith Labs. We're getting a second instrument upgraded to this, and this is sort of the latest and greatest product from Bruker that gives sort of the maximum sensitivity, which is really one of the critical things, but it also is very, very fast in terms of how many peptides it can sequence at a given time. We're still playing around with the chromatography to try to reduce the time to cram all those peptides into a shorter chromatographic separation. With a very, very fast instrument, you can do that. You get things down to like maybe 15 or 20 minutes, where a normal sample might take 60 to 90 minutes. You cram it down because there's in principle much less material there. You cram everything into a taller peak and you can still detect it. But there's still lots of optimization, I think, that can be done there. Okay, then at the informatics side, and I'm going to get to the lipidomics and then get to how we're trying to apply these to some biological systems. But on the informatics side, there's one last thing to say. So again, if you've ever been involved in a proteomics experiment, you know that you normally just search your data against a database of whatever organism it is that you're studying, and you get some results. That works for single cell proteomics data, but if you think about what's really going on in a single cell experiment, you've got sort of the bare minimum of peptides that you're trying to detect, and sometimes your signal to noise ratio is not as high as you'd like it. So there's a lot of noise in the data, and one really useful way to try to overcome that is sort of highlighted here. But what it does is what you do to achieve this is run a bulk sample. So by bulk, I mean you've got lots of material from you pool a bunch of cells, analyze it more sort of in the conventional proteomics approach, and then in parallel, you run all of your single cell samples. So you've got this sample with good signal to noise and a bunch of samples with really poor signal to noise. And you can get the software to look at those ones with the low signal to noise and look at the data from the high signal to noise sample and ask, do I find the same features? Do I find the same peptide here with the same mass to charge? And can I see it here and get better signal to noise on the fragments that are generated from that, so that I can use that to do a better database search? And indeed you can. So I won't really explain everything here, but if you look at the yellow or the white versus the green line, the white line is the number of proteins identified given different amounts of protein injected into the instrument. So starting down here at about 250 picograms. So we use that as a sort of a gold standard amount for what is expected, the amount of protein that's expected to be in an individual mammalian cell. Now there's big cells and small cells, and so there's lots of variation, but overall that's sort of where the field has landed. One cell, mammalian cell, contains about 250 picograms of protein. And so that's the low point here. The white line represents the data, or the number of proteins you identify if you use the conventional approach, where you don't use this high signal to noise sample, you just use the data as it is. If you include that high signal to noise sample and ask it to basically compare against that and find better data, you nearly double the number of proteins that you can identify from the same data. So this is using the same data. It's just you've left out, in this case, you've left out the high signal to noise data, and here you've included it in the analysis. So it's a little bit cheating, but not. You're not like manipulating the data. You're basically understanding a bit more about how the algorithm works and helping the algorithm find more reliable data that way. Okay, so then just a couple words about single cell lipidomics, and I'm going to have more to say about that as well. But there were a few things that enabled us to start doing single cell lipidomics as well. So again, the UNO device from TCAN for putting cells into wells becomes very useful. You have to make some modifications to the liquid chromatography system to eliminate losses and dead volumes in the system, using very high resolution liquid chromatography columns, more efficient ionization, and then same kind of instrument, but then also trying to better develop some of the software tools that are used to analyze the data at the end. Okay, I'm going to tell you a bit of a story about astrocytes. Oops, excuse me, because this has been Lucy's focus. So we started off this project wondering whether there would be differences in different sexes' response to virus infection. So this sort of sprung out of observations from SARS-CoV-2 and other viruses that we all know about, and how there are sexual dimorphisms in our responses to those viruses. We weren't really keen on trying to work with live SARS-CoV-2 itself. So we got help from Francois Jean's lab for Lucy to learn how to manipulate this other coronavirus, 229E. It's a coronavirus, so it's related to SARS-CoV-2, and it causes symptoms very similar to SARS-CoV-2 in mice. But we're working in human astrocytes in this case. Okay, so the sort of bulk approaches that we would normally use would really not allow us to drill into the different subpopulations within astrocytes and see whether sexual dimorphism might actually manifest differently in the different subpopulations of astrocytes that are known. Okay, so starting off just with a bulk analysis, and again, by bulk, I meaning we're taking a big pool of astrocytes and doing sort of a more conventional study without looking at individual cell differences. If you infect, or if you look at male versus female, in this case, not infected yet, there's lots of things you can pick out. You can find sex-specific differences that are known, things like that are on the Y chromosome, for example, only appearing in males, that kind of thing. Overall, there's quite a big difference, I would say, between male and female astrocytes, even though they're both in principle doing the same thing. if you look at those using a UMAP approach, they also separate. So you get these little subpopulations of male versus female. Sorry, I should say these are now individual single cell analyses, total of 320 cells represented here. There's clear subpopulations that are emerging, some that are way out in left field, it seems, and we're not really astrocyte biologists ourselves, but the general observation that there's multiple different subpopulations within astrocytes seem consistent. If we look at that from a more hierarchical clustering type of perspective, the cells break down into these different clusters that are colored differently according to protein expression patterns. The actual proteins that are represented here, not really going to go into those much, but just suffice to say that I should have mentioned at the top are female in the gold color and male in the whatever that is, bluey green color, and they don't all segregate out by sex clearly. Like these male cells are more similar to these female cells than these female cells are to those female cells. Should also caution that this is one male and one female donor at this point. So there's also the whole individual variation that might not necessarily all be linked to the sex of these donors. Okay, so then really this was just preliminary data to try to get to the point of doing infections. I will warn you, this is really where we're at now with the data. I'm going to show you some data on single cell lipidomics from these samples. We don't, or at least Lucy hasn't shared with me, any data on the single cell proteomics analyses of these cells. But the idea is we have male and female, infected and uninfected, and the MOI, or multiplicity of infection, of 0.2 for 72 hours for the actual infection, and then putting these cells into wells and doing the analysis. Again, I'm going to start off by showing you lipidomics. So in these cells, we're able to detect about 1,000 different seemingly lipid features. Of those, about 125 or so can be annotated as known lipids. The classes of the 1,000 or so are shown here. I won't really drill into that too much. The colors across the top here, you won't be able to read that, but that refers to the different male, female, infected, uninfected, but I'll expand a bit on that later on. If you look at these individual cells, again, just at the lipidomic data by PCA, or partial least squares discriminant analysis, you can see these subpopulations emerging. So these are the bulk cells. The uninfected cells seem more tightly sort of clustering, and the infected cells really, or the infection, I guess is the way to say it, really seems to screw around with the lipid profile in these cells quite dramatically. And I'll show you a couple examples of that. So some lipids are changing in both of the sexes. So there's one shown here. On the left are the original concentrations measured by mass spectrometry, and then when those data are normalized, they shift slightly, but the overall patterns remain the same. There's two lipids here that, well, in this case, are basically undetectable in the what's that? The female controls and the male controls, so that only when you get the virus infection can you actually detect these. Others, there's a baseline level in uninfected, but when you infect, they go up quite dramatically. You can find lots of some examples of lipids that are strongly affected in only one sex, so in this case, male. and don't seem to be all that impacted by the infection in female cells. You can find individual cells that seem resistant or at least, let's say, respond very, very differently to infection than most of the cells, even though they do still seem to be infected. And the overall homeostasis of the lipid profile in male cells seems to be far more affected than in female cells, which again is very early observation, and we don't really understand why. But certainly, even in female cells, the virus is also causing a big shift in all of the lipids that are present there. Okay, I'm getting close to being done. I want to wrap up just by saying this is really very hot off the press. We were talking this morning so I can understand the data a little bit better. So this is data that is unrefined at this point, but I think very intriguing. It is really enabled by our acquisition of this new mass spectrometer, Ultra 2, which is mostly helping on the proteomics side. We will be getting, hopefully in the next few months, a different new instrument for the metabolomics. So that will help a lot as well. One challenge that we have that I don't think we have a great solution for yet is how to deal with batch effects. And so you can use a 96 or a 384 well plate, but you could very well want to analyze more cells than that. And really what you'd want to do is randomize cells between plates and among conditions and everything. We don't have a good way with the devices we've got for putting cells into wells to do that in an efficient manner. So haven't gotten around that issue yet. There's still a lot of effort that has to go into driving down the amount of instrument time that goes into analyzing an individual cell. That really, I think, fundamentally will be the main limit to overcome to get the number of cells that we can analyze up. The bioinformatics always lags behind in a new field like this, and that's certainly true here. So there's a lot of work to do there. Some things to be learned from single-cell RNA-seq, but probably some new algorithms that will need to be developed as well. And I will say a little bit about network analysis. So what network analysis, so this is more now on the metabolomic side. And for those of you who haven't gotten involved in metabolomics, I would say it's remarkable how undeveloped the informatics is compared to proteomics, which itself is not that great compared to to sequencing methods. But in proteomics, we have a database to work against, right? All the proteins that we see should be represented in the possible proteins that could be encoded by the genome of whatever we're looking at. There's not necessarily that kind of link in metabolomics. And so interpreting the fragment spectra from a mass spectrometer run of metabolites is much harder. And so what an interesting approach I think that people are now taking is something called social molecular networking, where you look at, let's say this big one in the middle where you've identified it, you know what it is, it's well characterized, and maybe a couple other ones where you really know what they are. And then you've got all these other things around it that you have no idea what they are. But what you do know is that they have a similar mass to the thing that you know well. And more importantly, they have other similar characteristics, particularly similar kinds of fragmentation patterns. And so you can start to sort of build out a network of not interactions, but similarity, basically. So you've got this thing that you know, this other thing that you don't know, but it's very similar to this, so it must be very closely related from a structural point of view. And I want to end with a slide that Armando gave me. and ask you, well, I won't really expect you to answer this, but if I showed you these two little things and asked you what they were, you'd have no idea what they are. And why I like this slide is because it touches on another aspect of what my lab likes to do, or at least what I like to do with my lab, and that is hiking. So you might not know what those two things are, but if you see the whole picture and see everything that's around it, then you can realize that one of those things was a person and the other of those things was a rock. And this is sort of the concept that molecular social networking takes by looking at the larger picture rather than just only the data for that one particular molecule that you're trying to analyze. You can try to better understand the whole ecosystem. Okay, so I want to say one more thing. We are at this very early phase for single cell proteomics, and even more so for single cell metabolomics. It's still very much a numbers game, like how many can this person or this lab identify versus that lab identify. And one thing that I'm really trying to push, I think our whole lab is trying to push, is that we really need to get away from that numbers game, at least in terms of the things that we can identify, and talk more about the things that we can quantify. Because you can identify lots of things in a single cell RNA, single cell proteomics experiment, but if you haven't quantified them across all your cells, they're useless. So you need to focus on what you can quantify at the protein or the lipid level across most of your cells in order for that data to be useful. And we have a long ways to go in terms of sort of finalizing best practices, and particularly around how we analyze the data. All right, so I always end my talks by thanking my kids. And many of you know, well, actually, I'll go back one. Many of you know that I have two kids, and I've done a lot of hiking with them, and very much enjoyed that. These are them sleeping in a snow cave. But about 6 or 7 months ago, I got married, and my new wife, Dana, also had two kids. And so now I have 4 kids who are constant sources of inspiration for me. The other reason I put this up here is to remind myself and everybody listening that as much fun as science is, there's more to life than science. So thanks very much for listening, and hopefully I can answer any questions if you've got some. Yeah.
Hey, thanks so much for that presentation. I'm wondering about the limitation on the throughput. Is the limitation purely an issue of volume surrounding the cell? In other words, if you had a magic wand, you could make the volume smaller, with something else get in the way, like instrument time per on each sample kind of thing.
Yeah, I think it would be folly to say that something else wouldn't get in the way. I still maintain right now the main limit is not even so much the volume that you can get the cell into, although I think we've got very good data that the smaller you can drive that, the better your data will be. But we still are needing dozens of minutes of mass spectrometer time, at least, to analyze one of those cells. And mass spectrometers are expensive and hard to run, so you can't just scale that up so easily. So if we can drive down the amount of time dedicated to one analysis in the instrument, that will increase our throughput. But then I would say the next limit on throughput would be the computational power needed to analyze that data. And that's, I think that people in my lab would probably agree that it's fair to say that that limit at the computational power is not far off of the limit of the mass spectrometer time as well, because it still is taking a long time to analyze the data. Robin.
What kind of platform are you doing that computation on? Because like with genomics, we're doing thousands of cells and sets, but doing them in high performance computing environments.
Yeah. So we are using we use everything from laptops to HPC systems as well. And obviously HPC systems help you do things faster. So there's a few issues there. One is the software kind of lags behind to really take advantage of massively parallel compute. So there's some limits to overcome there that are more on the software design side than the availability of the actual compute.
Bioinformaticians love these problems.
They do. Agreed.
Thanks a lot. Can we label the surface lipids on cells so that we can just start looking to see what's there? Because that's fundamentally interesting.
Yeah. So in principle, yes. The practical limitation is still the amount of material there. So maybe Lucy has done something that I'm not aware of, but like taking all of the proteins into it from a cell and getting them into the instrument, that is really only allowing us to identify maybe a 20 to 25% of the proteins that are present there. If you start looking at a subcellular section, then we're not going to be able to identify much, but I think you could do it. And in the right, for the right kind of question, probably you could get good data. You wouldn't get like 1,000 membrane proteins, but you might get 50 or 100. And if the question's right, it might be good.
I was wondering about the lipid.
Oh, the lipids. So maybe someone who knows more about this than I do would have a solution to this. Right now, we're doing an organic extraction on the cell, and I don't see how you would do that and only get the external lipids, like the plasma membrane. Maybe there's a good way to do that with some kind of labeling like you're getting at. The problem that we're always running into, and I kind of touched on it a couple times, is the more manipulations you do, the harder it is to detect everything. So if you did some kind of, you're thinking of like a chemical label of some sort. Yeah, I mean, I guess you could. And we started with lipids among the metabolite classes. We started with lipids because they're relatively easy to work with and separate from everything else because they're so generally so polar. So if you did some kind of chemical label on the surface of the cell, you probably could get something there. Armando, do you want to comment on that? Like, are we right at the limit of detection that you wouldn't get much, or what do you think? I guess what I would say is if you've got a experiment or a question in mind, like a specific question, I presume you didn't just pull this out of your head randomly. But if there's a specific situation, it'd be easy enough to try it. And one of the challenges with chemical labeling is that there's very, very few chemical reactions that go to completion. So you might get like 30% labeling, and then your sensitivity suffers greatly that way. But if you got a question, let's talk about it. Okay. Okay. I guess that's it. Thanks for your attention, everyone.